History of biotechnology. Important contributions, and here is the scientist. First in it started with the 1902 Abelard, first attempt of plant tissue culture, father of plant tissue culture. Then Henning's first attempt to culture embryo of selected cruciferous. Another is Knudsen, a symbiotic germination of orchid seed in vitro. In vitro means that. Robinson, in vitro culture of root trips, then in laboratory. Okay. Then 1925, that is Liebach, use of embryo culture techniques in interspecific cross of linseed, that is linum. Then Goethrit, in vitro culture of cambial tissue of few trees and shrubs, although failed to sustain cell division. Then White, White, successful culture of tomato roots in 1934. Then 1939, there are three scientists as I previously discussed: Goethert, White, Knobcott, successfully establishment of continuously growing callus culture. Then Goethrit, in vitro culture of cambial tissue of ulmus to study the adventitious shoot formations. Then underline is very much important, you have to remember. Van over Beek, use of coconut milk containing a cell division factor for the first time to culture datura embryo. Then Ron Skoog is also in vitro adventitious shoot formation in the tobacco. Then the Morel and Martin use meristem culture to obvious virus-free dahlias. Then first application of micrografting by the Morel and Martin. Then a Korenberg and his co-workers studied on in vitro synthesis of DNA. Now Skoog and Miller discovered the regulation of certain organ formation by changing the ratio of auxin and cytokinins by different concentration of that. They have described this. Then Maheshwari and Rangaswamy in 1958, they have somatic embryo and in vitro from the nucleus of citrus ovules. Then Reinach and Stewart, regeneration of embryos from callus clumps and cell suspension of carrot. Then Kanta, then Murashing and Skoog, that development of MS medium, nutrients. Then Guha and Maheshwari. Guha and Maheshwari, as I said. Argovind Khurana, H.G. Khurana, awarded Nobel Prize for deciphering of genetic codes, deduced the structure of gene for each tRNA. That is also a contribution of Khurana and parties. Then you have to remember this protoplast fusion, first achievement by Power, and then Temin and Baltimore. This discovered, he has discovered the presence of a reverse transcriptase RNA directed DNA polymerase which has ability to synthesize cDNA using mRNA as a template. That is given by this, and they have written books also. Now Smith has discovered first restriction endonucleases, and it was purified name as Hind, Haemophilus influenza, in 1970. Now 1971, this Nathan's preparation of first restriction map using Hind2 enzyme to cut circular DNA or SV40 of to 11 specific fragment. Then Carlson and Power, further this Power interspecific hybridization by protoplast fusion. And then these are the various scientists you have to remember. Here is Carl Mollis in 1983 conceived the idea of polymer chain reaction and chemical DNA amplification process. Then Alice Jeffrey, development of genetic fingerprinting techniques for identifying individuals by analyzing polymorphism at DNA levels. These are the various scientists and their contribution in 1987, isolation of Bt genes for bacterium. Then Williams, development of RAPD techniques. Not only a Williams, Wells and McLeod. A&D याद रखना है. Human Genome Project is completed in 2001. Specific sequence of human genome successfully completed. And lastly, these are the various scientists who have contributed towards the biotechnology and plant tissue cultures as well as human biochemistry too. Okay, now we will see the certain terminology which are useful in this topics. Okay, first is culture. What is culture? If it is asked in the examination, then you have to write growing of cell, tissues, plant organ, whole plant in nutrient medium under aseptic condition. Depending upon the explant source, it can be named as various culture. Okay. For example, organ culture, cell culture, aseptic culture, suspension culture, batch culture, continuous culture. Okay, these are the various culture. Depending upon organ, you will find a culture of isolated plant organs such as root tip, shoot tip, leaf primordial, immature plant of flowers that can culture under the nutrient medium under aseptic condition depending upon the explant which is utilized. Then cell culture means culturing of the single cells or small group of similar cells. Then what is aseptic culture? Aseptic means arising of culture from tissue and organ after elimination of bacteria, fungi and microorganism. Now what are the suspension culture? Suspension culture means culturing of cell, okay, or cell aggregates in the liquid medium. Then batch culture. Batch culture, same cell suspension grown in fixed volume of liquid medium, that is called batch culture. And continuous culture means what? A suspension culture continuously supplied with the nutrient by continuous flow of fresh medium. The volume of culture medium is normally constant. This is all about the certain terminology. Now as we say that this culturing of any plant organ through certain medium. So what is nutrient medium? Nutrient medium, it may be a solid or liquid or combination of nutrient and water, usually including several salt, carbohydrates in the form of sugars and vitamins such as medium is called basal medium. Okay. The basal medium may be supplemented with the growth hormones, occasionally with other defined or undefined substances. Okay, this is all about the nutrient medium. Now there are major plant hormones are useful in the plant tissue cultures or culturing of the any plant. For example, auxin, a class of hormones which it has a function like cell elongation, apical dominance, and in culturing it will find root initiation. So the different concentration of these auxins that gives you a root initiation. So these are the examples: naphtalic acetic acid, indole acetic acid, and 2,4-D. Okay, this is auxins. Now what is cytokinins? Cytokinins, it is also a class of growth hormones, but it has a functions which cause cell division, shoot differentiation, breaking of apical dominances. What are that? Example, etc. like kinetin and zeatin. Explant a piece of tissues used to initiate tissue culture or removing shoots from the callus, separating individual shoot from proliferating mass of shoots. This is the function of cytokinins. Now this is all about the nutrient mediums and hormones. Now we will see the certain terminology after the growth media initiations. What is callus? A tissue arising from disorganized proliferation of the cell either in culture in nature, or we can say mass of cells. Now subculture, aseptic transfer of part of culture to fresh media. Another terminology, phase time, the time interval between two successive subculturing of the tubes or media or any other mediums, that is called passage time. Now what is clone? Clone, as we know, that's similar to single individuals. A population of cell derived from single cell by mitotic divisions, a propagation of mitotic divisions, plants derived from single individual plants through vegetative propagation. Now this is the terminology of clones. Now micropropagations. These words comes in PTC. Okay, plant tissue culture. Production of miniaturing plant material, somatic embryo or plantlets in large number by vegetative multiplication through growth hormone regenerations. Foremost important, what is totipotency? The ability, inherent property of cell or tissue to give rise whole plant irrespective of their plody level and form of specialization. Means plant have a ability to regenerate new ones. Okay. So the totipotency means the ability of inherent property of cell or tissue to give rise to whole plant irrespective of plody levels and form specializations. Okay. Then meristem. What is meristem? A group of actively dividing cells from which permanent tissue systems such as root, shoot, leaf, flower, etc. derive. Now another terminology, embryoid and somatic embryos. Means non-zygotic embryos formed in the culture. Now what mean by organogenesis? It is the types of morphogenesis which result in the formation of organ or origin of shoots, offshoot roots, the floral organ from tissue culture or suspension culture. This is organogenesis. Now regeneration. Regeneration means it is in tissue culture, it is used for development of new organs, plantlets from the tissue culture from a bud. Now embryogenesis. What is embryogenesis? The process of embryo initiation, origin of plants in the development pattern that closely resemble to the normal embryo development from fertilized egg or ovum. In vivo, a Latin word literally means in living, applied to any process occur in the whole organism under field condition where there is no control over the environmental conditions. Okay, this is called as in vivo. What is in vivo? A Latin word literally means living, applied to any process occur in the whole organism under field condition where there is no control over the environmental condition. Now what is in vitro? A Latin word literally means in a glass or living in a test tube applied to any process carried out in the sterile culture under the control condition in the laboratory. In vitro means in laboratory, in vivo means in the field condition. Okay. Now another terminology is amplification. Generally amplification done in the PCR. Okay, polymer chain reaction. So you have to remember creation of many copies of segment of DNA by PCR or duplication of genes within the chromosome segment. That is called amplification. Now parasexual hybridization. What is parasexual? Yes, hybridization by non-sexual method, that is protoplast fusion. Okay. Now, hybrid. In many examination, that is asking what mean by hybrid and hybrid, differences between hybrid and hybrid. So you have to remember this plant or cell which is cytoplasmic hybrid produced by the fusion of protoplast, cytoplast. Okay. Now the word come protoplast. What is protoplast? A single cell with their cell wall stripped off a cell without cell wall. Now another same terminology comes in this hybrid, that is cytoplast. Protoplast, a nucleus enucleated protoplast. Okay, this is all about the what? PTC biotechnology. Heterokaryons, homokaryons, synkaryon. These are the terminology. Hetero means different. A cell in which two or more nuclei of unlike genetic makeup are present. Homo means karyon, a cell with two or more nuclei or similar genetic makeups. Okay. Synkaryon, synonymous. That hybrid cell produced by the fusion of nuclei in heterokaryon. Heteroplast, cell containing foreign organelles. Or most important for this course, what mean by genetic engineering? As we know that genetic engineering deals with the organism or manipulation in the genetic structures or changes in the genetic structure of that for the development or for human use. Okay. Or further improvement in that structure. So manipulation of genetic architecture of an organism of DNA level, it means molecular rDNA technology. Okay. What mean by rDNA? Recombinant DNA. The DNA which contain gene from different source can combine with the DNA of any organism. Some DNA from a plant, insect. Okay. That can manipulate in between the species. Okay. Now what is transgenic plant? Simply you can write plant which contain foreign DNA.